Hello, everyone. It's my pleasure to present a talk entitled A Novel Accomplishment of Chemical Artificial Intelligence Using Chemical Waves to Discriminate Acoustic Frequencies. First of all, the outline of my talk. Why am I developing chemical AI? Well, I give you an answer by mentioning the necessity to face the complexity of the 21st century challenges. Then I will give an answer to the question, what is chemical AI? And I will present an example. It is a recent achievement in our laboratory. It's a bio-inspired Chemo-Acoustic system. And finally, I will conclude by presenting some perspectives of chemical artificial intelligence. Let's get started. Currently, we are spurred to face global challenges. A collection of global challenges is included in the 2030 agenda, compiled by the members of the United Nations. Reaching the goals of this agenda means dealing with human beings and their societies, the world economy, the urban areas, the natural ecosystems, and the climate. These systems are instances of what we call nowadays complex systems. So it's evident that whenever we face global challenges, we need to deal with complex systems. However, we experience seemingly insurmountable obstacles whenever we deal with complex systems. And these obstacles outline the epistemological complexity. A contribution comes from computational complexity. Many computational problems regarding complex systems are solvable but intractable. It means when we face these problems that have large dimensions, then we cannot determine their exact solutions in a reasonable time, even if we use the fastest supercomputers in the world. A second contribution to the epistemological complexity comes from the recognition of variable patterns that are emergent properties of complex systems. We still need to formulate universally valid and effective algorithms for recognizing every type of variable patterns. A promising research line to face the epistemological complexity is natural computing, an interdisciplinary research line that draws inspiration from natural phenomena to formulate new algorithms, propose new materials and architectures to compute, and new methods and models to understand complex systems. Based on the idea that any distinguishable physicochemical state of matter and energy can be used to encode information, and every natural transformation of these states is a kind of computation. Within natural computing, we distinguish two programs. In the first, scientists exploit the physicochemical laws to make computation. By knowing that any physicochemical law describes a forward causal event, and any forward causal event can be conceived as a computation, because the causes become the inputs, the effects become the outputs, and the law governing the transformation becomes the algorithm of the computation. In the second program, scientists mimicked information gathering and utilizing systems belonging to living beings. So we might mimic how cells compute, or how nervous systems, immune system, and societies of agents compute. Chemical artificial intelligence derives from natural computing. It's an interdisciplinary research line that tries to answer the following heuristic questions. Which performances of biological intelligence can be mimicked through inanimate chemical systems in water, in a liquid solutions, which is the characteristic phase of life? In this graph, I show the knowledge map of chemical artificial intelligence. It's a three-dimensional plot projected on a plane. Along the x-axis, we have the chi domains that are molecular and supramolecular chemistry, system chemistry, and multiscale chemistry. With multiscale chemistry, I refer to all those chemical systems that manifest self-organizations at different spatial scales and multi-level causality when appropriate macroscopic thermodynamic forces are applied. Such chemical systems, maintained in out-of-equilibrium conditions, originate the well-known dissipative structures and forms of active matter. Multiscale chemistry is required to mimic the structural and functional performances of multicellular biological organisms that exhibit multiscale competencies. It means biological organisms, multicellular biological organisms, process different kinds of information at different spatial scales. Let's go back to the knowledge map of Chi. Along the y-axis, we have the Chi paradigms that are biosensor, brains, and effectors. And finally, in the third dimension, represented by the saturation degree of the green color, we have the biological intelligent competencies that range from sensing to perceiving, from computing to planning, from working to acting. Some of these biological intelligent competencies have been already implemented. They are indicated in black, but there are some others that have not been implemented yet. They are indicated in white. An ambitious goal of chemical artificial intelligence is the development of chemical robotics. The chemical robot is thought of as a confined molecular assembly that reacts autonomously to its environment because it has sensors collecting data about the environment and internal state of the robot. It has artificial neural networks to process the sensory data, make decisions, and trigger the effector modules that can act upon the environment. The intelligent activities of a chemical robot should be sustained by a metabolic unit. Chemical robots should be easily miniaturized in order to be implanted in living beings and interplay with cells, organelles to perform biomedical actions. In other words, chemical robots should become auxiliary elements of our immune system. However, we might also think of deploying these chemical robots in the environment to safeguard the environment and face problems related to energy and food supplies, as enticingly alleged by the futurist Kurzweil. And finally, the development of Chi might have a curious side effect. It will assist human efforts to unveil the mysterious event of the appearance of life on Earth. The appearance of life on Earth can be described as a staggering phase transition in how chemical systems, from a completely abiotic world to a world where the first chemical system capable of exploiting matter and energy to handle information and pursue goals appeared. And this staggering event occurred roughly 4 billion years ago. And now, as I mentioned at the beginning of my talk, today I'm going to present a recent achievement in our laboratory that is a biospire Chemo-Acoustic system based on the nonlinear phenomenon of chemical waves that are autocatalytic reactions propagating through excitable media. For instance, electrochemical waves propagate in the form of action potentials along the axon of a neuron for information transfer, and electrochemical waves propagate in the form of spindles in our cerebral cortex to consolidate and integrate memories during our sleep. The Belousov-Zhabotinsky reaction, that is a metal-catalyzed oxidative bromination of malonic acid in aqueous acidic solution, gives rise to outstanding examples of chemical waves, as you can see from this movie. And since the behavior of the busy reaction is strongly affected by mixing, we explore the response of its chemical waves to mechanical oscillations. In this picture, I show the experimental setup. On a petri dish, polystyrene petri dish, we pour a T layer of the busy reaction, and then we apply vertical vibration with frequencies in the range of the acoustic range. We discovered that the busy reaction shows sensitivity to mechanical vibration in the range of 2010 Hz, and we discriminated 7 acoustic bands in this range. And now I show the experimental results. First of all, I show the behavior of an unperturbed busy reaction. We see target patterns. They can be either dense or sparse, and we also see spontaneous phase wave moving from the periphery towards the center of the dish. What happens if we apply frequency in the range 2650? Well, we still see target patterns and spontaneous phase waves. However, the mechanical vibration make the front waves of the target patterns much more irregular. What happens if we apply frequency in the range 650-500 Hz? Now we have a pacemaker of advection-induced phase wave in the center of the dish. And the advection-induced phase wave propagate anisotropically towards the periphery, generating clover-type shapes. In the periphery, we still see the formation of spontaneous phase wave and target patterns with irregular front waves. What happens if we apply frequency in the range 500-200 Hz? Now we see a vibrating annulus in the middle of the dish, and the advection-induced phase wave are generated in this region. Then the advection-induced phase waves propagate towards the center, and finally, they spread slowly in the rest of the solution. What happens if we apply frequency in the range 295 Hz? Now we see two vibrating regions, one in the center and the other in the periphery, separated by a steady nodal region. The advection-induced phase waves are generated in one point of the two vibrating regions. Then the advection-induced phase wave propagate in the rest of the solution, engulfing the nodal region. What happens if we apply frequency in the range 95-50 Hz? Now we see a grid of cells. That is an instance of the so-called Faraday pattern. We have standing waves. The amplitudes of the oscillations in the periphery are larger than in the center. The advection-induced phase waves are usually generated in the center, and they propagate radially towards the periphery. What happens if we apply frequency in the range 50-15 Hz? Now the grid of cells is uniform. The amplitudes of the oscillations are uniform everywhere. The dimensions of the cells increase by lowering the acoustic frequency. And the advection-induced phase waves are generated in one point of the periphery of the dish, and they move quickly towards the opposite side, sweeping the entire solution. What happens if we apply frequency in the range of 15-10 Hz? Now the cells are so large, they fragment the front waves of the advection-induced phase wave, and these waves appear irregular. The velocity of the advection-induced phase waves is apparently dependent on the acoustic frequency, as you can see in this plot. The lower the frequency, the faster the propagation of these waves. The interpretation of this dependence was possible by describing the entire solutions through the network of cells model. In other words, the layer of the solution is granulated in microreactors that exchange chemicals through diffusion and advection. The linear dimensions of these cells is of the order of 7, the elementary cells is of the order of 7 micrometers. And the vertical mechanical vibration affect the chemical coupling of these cells. The lower acoustic frequencies guarantee a more extended chemical coupling, the formation of larger clusters, and hence a faster propagation of the advection-induced phase waves. This system is an instance of multiscale chemistry, because the dissipative spatiotemporal patterns I showed you before are due to the vertical vibration of the Petri dish at the macroscopic level, micrometric open reactors that exchange chemical reagents through diffusion and advection, and of course, these cells involve the mesoscopic level, the micrometric level, and finally, we have the busy reaction at the molecular level. 3 are the key outputs for discriminating the 7 acoustic bands in the 2010 Hz range. The first key output is, of course, associated with the spatiotemporal patterns we observed. These patterns involve target patterns, spontaneous phase wave, and advection-induced phase wave. This plot reports the frequency of these spatiotemporal patterns as a function of the acoustic frequency. And we observe that the frequency of these spatiotemporal patterns are from 3 to 5 orders of magnitudes smaller than the acoustic frequency. A second key output is the dependence of the advection-induced phase wave velocity on the acoustic frequency that is described by this bioexponential function. And finally, the third key output to discriminate the 7 acoustic bands is the dependence of the Faraday wavelength on the acoustic frequency, described by this function and shown in this plot. We can make some analogies between our bio-inspired chemoacoustic system and a human ear. In the human ear, the cochlea is the transducer of mechanical energy into electrochemical energy due to the presence of ear cells. And in the cochlea, we have a topographic representation of the acoustic waves along the asymmetric basilar membrane. The lower frequencies are detected at the apex of the basilar membrane, whereas the higher frequencies, the highest frequency, are detected at the beginning of the same membrane. In our bio-inspired chemoacoustic system, the Biot reaction is the transducer of mechanical energy into chemical energy, and it offers peculiar spatiotemporal patterns as representation of different acoustic bandwidths. And now I move to the conclusion by presenting some perspectives about chemical artificial intelligence. First of all, chemical AI and chemical robotics can help humans explore and colonize the poorly investigated space so far, that is the molecular world. Decolonization of the molecular world will provide tools to effectively combat poverty, clean up the environment, overcome diseases, and extend human longevity. Trying to obtain intelligent chemical systems from scratch can help understand intelligence and life. We are aware that chemical AI and chemical robotics are in their infancy. However, it's worthwhile pursuing their development. And to be productive, we need a fruitful interdisciplinary collaboration between chemists and specialists of other disciplines, as it usually happens in translational chemistry. Finally, I want to mention that there is an open issue devoted to chemical artificial intelligence for this MEDPI journal. You are invited to contribute. And I want to acknowledge my collaborators, specifically for the implementation of the bio-inspired chemoacoustic system. I want to mention Professor Epstein and Professor Dolnik. I want to acknowledge my students, specifically for the bio-inspired chemoacoustic system. I want to mention and thank Laura Tomassoli, and I thank the European Union and my university for the financial support, and all of you for your attention.